Hello everyone, welcome to Bioscope, where we decode the language of life. In today's video, we will explore DNA sequencing, the powerful technique that allows scientists to read the exact order of nucleotides in our genetic code. From understanding hereditary disease to advancing personalized medicine, DNA sequencing has revolutionized biology and medicine. Stay tuned as we break down the methods, application, and future of this fascinating technology in a simple and clear way. Before starting, here is a quick recall for you, that is DNA, gene, and chromosome. Can you please match these three things with the lower boxes to recall exactly what DNA, gene, and chromosome is, or the basic differences among these three is? You have 10 seconds for that. Okay, now let's check the answer. Here you can get a clear view of what gene, DNA, and chromosome is. For that, you can also go to my DNA video to understand these things. Now let's start what DNA sequencing is. DNA sequencing is a laboratory process of determining the exact order of nucleotides, that is the adenine, thymine, cytosine, and guanine in a DNA molecule. It is fundamental to genetics, molecular biology, biotechnology, and medicine, allowing researchers to decode the genetic instruction that guide all living organisms. This technique is based on some basic principle. That is, DNA sequencing works by reading DNA sequence of nucleotides in a DNA strands. And the modern sequencing methods rely on some basic things, that is DNA polymerase enzyme to synthesize new DNA strands, detection of incorporated nucleotides either by chain termination, fluorescence leveling, or real-time monitoring. Then the conversion of signals into a readable digital sequence. Now let's talk about the development of the process of DNA sequencing. In 1970s, the first sequencing attempts was done by Maximum Gilbert method, and then in 1977 Sanger's chain termination method was developed. Then in 1980s the automated Sanger sequencers improved speed to the throughput. In 2000s, the next generation sequencing is developed. Platforms like Illumina, Roche 454, and Solid emerged, allowing massive parallel sequencing. And nowadays, third generation sequencing is done with the help of real-time single molecule sequencing with PacBio SMRT Oxford Nanopore enable ultra long reads. If we talk about the major sequencing methods, then it includes Sanger sequencing, next generation sequencing, and third generation sequencing or SMRT. Let's have a look on each of the techniques in a nutshell. In the Sanger sequencing, the technique uses dideoxynucleotides or DDNTPs to terminate DNA synthesis at specific base. It includes some processes where first DNA template is denatured, then primer binds to the template, DNA polymerase synthesizes new DNA strand with the presence of normal DNTPs and fluorescently or radioactively labeled DDNTPs. Then resulting fragments are separated by capillary electrophoresis, and lastly a laser reads the fluorescent signal to determine the sequence. This process has some advantages, like it has high accuracy, suitable for small scale sequencing, but its low throughput or expensive for large genome makes it some limited application of itself. The next thing is NGS or the next generation sequencing. It is based on a principle that says massively parallel sequencing or millions of short DNA fragments are sequenced simultaneously. Some platforms, for example Illumina, helps in the sequencing by synthesis with fluorescent reversible terminators. In Roche 454 pyrosequencing, it detects light emitted upon nucleotide incorporation. In solid sequencing, sequencing is done by ligation with color space detection. The advantage of this technique is high throughput, cost effective for whole genome sequencing, but the limitation includes short read lengths, complex bioinformatics analysis. Next is third generation sequencing, which is based on the principle that single molecule real time SMRT or nanopore based methods sequence DNA without amplification. The key technologies include PacBio SMRT sequencing, which states that DNA polymerase incorporates fluorescently labeled nucleotides in real time. In Oxford nanopore sequencing, it measures changes in ionic current and DNA passes through a nanopore. The advantages of this method is very long reads can be done. It detects epigenetic modification, but the limitation is it has high error rate than Illumina. The DNA sequencing has many applications. For example, in genomics, we can do whole genome sequencing or competitive genomics. In medical research, it can help to identify genetic mutation in disease. It can personalize a medicine, or it can help in the research of pharmacogenomics. In forensic sciences, it helps in the DNA profiling for identification. In evolutionary biology, it helps studying genetic relationships between species. In agriculture, we can use this to improve the crops and in breeding programs. In metagenomics, it helps in analyzing the microbial communities in environmental samples. Advantages of modern sequencing is it is highly sensitive and it is accurate. It has the ability to sequence unknown DNA with prior knowledge, and it is rapid turnaround time for large projects. But it has some challenges and limitations also, because it requires expensive equipment and reagents. It generates massive database needing advanced bioinformatics tools. The short read methods struggle with repetitive regions, ethical concerns regarding privacy of genetic data, but is had some major future direction. That is, it is ultra fast real time sequencing for clinical diagnostics, portable sequencer for fieldwork and point of care testing, AI assisted analysis for interpreting massive genomic databases, and integrating sequences with CRISPR based diagnostic for rapid pathogen detection. So that's all for this video. I hope you understand. If you like the video, please hit the like button and subscribe for more such videos. Thank you for watching.